Today, we'll be doing a Mindmap on proteins and its applications, which is a continuation of what I discussed in my Mindmap on molecular biology. Proteins can be classified as either globular or fibrous, with the following comparisons. Globular proteins have rounded globular shapes, while fibrous proteins are long and narrow. The role of globular proteins is generally for functional purposes, for example catalysis, while fibrous proteins is for more structural purposes, for example support. Globular proteins are generally soluble in water, while fibrous proteins are insoluble. Globular proteins have irregular amino acid sequences, while fibrous proteins have repetitive amino acid sequences. Globular proteins are also sensitive to changes in the environment, such as changes in temperature and pH. Fibrous proteins tend to be less sensitive in comparison. Here are some examples of the roles proteins play and their categorization into these two groups. Firstly, proteins form enzymes which catalyze specific reactions. This role is performed by globular proteins, with an example being rubisco, a major enzyme in carbon fixation. Fibrous proteins give tensile strength in tissues such as skin, tendons, ligaments, and blood vessels. An example of a protein responsible for this is collagen. Proteins have a role in muscular contraction, which is essential for locomotion and transport within the body. The globular proteins actin and myosin play this role. Proteins also play important roles in membrane transport, where proteins allow for movement of substances within and across membranes. These forms of transport include diffusion, active transport, and electron transport, with an example being the fibrous protein ATP synthase. Proteins in blood plasma are essential for blood clotting, as is the case for the globular protein thrombokinase, which acts as a clotting factor. The fibrous protein tubulin is also essential for the cytoskeleton of cells, which is what gives animal cells their shape. Tubulin also forms microtubule polymers, which is needed for the splitting of chromosomes in cell division. These are just some examples of globular and fibrous proteins and their functions. You'll come across various proteins across the biology syllabus, so it will be useful to be able to differentiate fibrous and globular proteins so that you'll be able to identify them when you see them. And it will also be helpful to memorize some examples as well. As discussed in my Mindmap on molecular biology, proteins are made up of amino acids. Amino acids undergo condensation reactions to form peptide bonds with each other, characterized by this peptide linkage. A straight amino acid chain forms the primary structure of proteins. However, proteins will usually form more complex structures. This can be secondary structures where the chain begins folding and turning. This is due to hydrogen bonds that form between the amine and carboxyl groups of non-adjacent amino acids. This allows for increased stability. When proteins go on to form tertiary structures, they begin to develop three-dimensional shapes, where bonding and interactions begin existing between the R groups of the amino acids. Quaternary structures form when there's an interaction between multiple polypeptides and potentially prosthetic groups. Prosthetic groups are inorganic compounds in the quaternary structures, with an example being the heme group present in hemoglobin, which gives the protein the ability to bind to oxygen. Fibrous proteins tend to be restricted to forming primary and secondary structures, while globular proteins form tertiary and quaternary structures. However, there are exceptions, with an example being collagen, which we touched on. Collagen is made up of 3 polypeptide chains, which folds to form a triple helix quaternary structure. One important thing to note is that the structures a protein forms is already determined by the amino acid sequence, which determines the bonds and interactions that occur within the protein, and hence its foldings and conformations. A very important application of proteins is enzymes. Enzymes are proteins that function as biological catalysts. They speed up or alter chemical reactions, but remain chemically unmodified after the reaction. This means that enzymes can be used again. Enzymes catalyze reactions using their active sites, where reactants, known as substrates, collide with the active site and bind to it to form an enzyme-substrate complex. Once formed, the enzyme can catalyze the reaction, in this case the breakdown of maltose to glucose. Maltase enzyme catalyzes the breaking of the glycosidic bond in maltose to release 2 glucose molecules as products. As you can see, the enzyme remains chemically unchanged after the reaction, where it can form an enzyme-substrate complex with maltose again to catalyze its breakdown. Due to this, one characteristic of enzyme is that it is often only required in minute amounts to cause a big reaction. What I just described is an example of a catabolic reaction, where a substrate is broken down to simpler products. Enzymes function for 2 main purposes. the breakdown of complex substances to simpler substances, also known as catabolism, and the synthesis of complex substances from simpler substances, also known as anabolism. Both of these occur via metabolic pathways, which may involve a series of chemical reactions and intermediate products between the initial reactant or reactants and the product or products respectively. How enzymes catalyze chemical reactions is by providing an alternative pathway with a lower activation energy. Continuing from my example of maltase breaking down maltose to glucose, in the absence of maltase enzyme, maltose, the reactant, will have to raise its energy to this certain amount to possess the activation energy needed for the catabolic reaction to take place. Activation energy is defined as the minimum amount of energy the reacting species must possess to undergo a specified reaction. In the presence of maltase, however, the enzyme lowers the activation energy for the reaction. This speeds up the reaction where maltose is now broken down to glucose faster, and it requires less energy. This graph shows the energy changes for a catabolic reaction, where the products of enzyme-catalyzed reactions are always at a lower energy level than the reactants. The opposite is true for anabolism, where products formed are always at a higher energy level than the reactants. This is due to the nature of catabolism being that of bond breaking, which releases energy, while anabolism involves bond formation, which requires energy. However, regardless of the reaction, enzyme works in the same way by lowering the activation energy needed for the reaction to occur. One important thing to note is that activation energy is always measured with respect to the energy level of the reactant. There are multiple factors that affect enzymatic activity. One of them is temperature, whose effect can be plotted by this graph. At initial low temperatures, increases in temperature does not have substantial effects on the rate of enzymatic reaction, as the temperature is still too low and there is low activity in the enzyme-substrate solution. However, past a certain threshold, increasing temperature begins to cause significant increases in the rate of enzymatic reaction. At this portion of the curve, a 10 degrees centigrade rise in temperature can increase the rate of enzymatic reaction by up to 100%. The reason why temperature has this effect on enzymatic activity is because when the temperature of the system is increased, enzymes and substrates begin moving faster in a solution. This increases the rate of collision between enzymes and substrates, hence increasing the rate of successful collision to form an enzyme-substrate complex. The peak of the graph shows the optimum temperature at which a particular enzymatic reaction occurs. After this point, increasing temperature causes the rate of reaction to drop sharply as enzymes begin to denature. Remember that enzymes are proteins with bonds holding them together. Therefore, when the temperature gets too high, these bonds break and the enzyme loses its shape and thus active site. Enzymes are considered to be denatured at this point, where the lack of an active site means that enzyme-substrate complex can no longer be formed. The increased velocities of enzymes and substrates in solution, leading to an increased rate of enzymatic reaction, can also be explained in terms of activation energy. Due to the increased rate of collision between enzymes and substrates, more reactants are able to reach the activation energy, which increases the rate of reaction. Another factor that influences the rate of enzymatic activity is pH. Every enzyme has an optimum pH range, which tends to be very narrow. I'll use digestive enzymes to explain this factor. In the human digestive tract, there are multiple environments with varying pH conditions. Digestive enzymes have an optimum pH range which corresponds to the environment in which they act. In the stomach, which is acidic in nature, enzymes such as pepsin will have an optimum pH of around 2. In the mouth, which is about neutral, salivary amylase will have an optimum pH of around 7, while in the small intestine, which is slightly alkaline, enzymes such as trypsin will have an optimum pH of around 8. The narrow pH range means that outside the range, the rate of enzymatic reaction drops drastically. This is due to the denaturation of enzymes, where extreme pH levels outside the optimum range causes the active site of enzymes to deform, preventing the formation of enzyme-substrate complex. The last factor that affects enzymatic reaction is substrate concentration. As substrate concentration increases, there is an increase in the rate of reaction because with more substrates in the solution, the frequency of collision between enzymes and substrates increases. This increases the frequency of successful collision to form enzyme-substrate complex, and as such increases the rate of enzymatic reaction. However, past a certain point, any increase in substrate concentration is matched with a diminishing increase in the rate of enzymatic reaction. This continues until the curve plateaus, where increasing substrate concentration no longer has any effect on the rate of reaction. This later effect of increased substrate concentration on the rate of reaction is because the concentration of enzymes in the reacting solution remains the same. When a substrate binds to the active site of an enzyme to form enzyme-substrate complex, the enzyme is considered to be occupied, where it cannot bind with another substrate until the reaction is completed. Therefore, when substrate concentration increases, there are now more active sites occupied at any point in time as the rate of formation of enzyme-substrate complex increases. After a certain point, assuming that the concentration of enzymes remains unchanged, increasing substrate concentration results in smaller increases in the rate of reaction as there's a decreasing availability of unoccupied active sites for substrates to form enzyme-substrate complexes with. On this idea of occupied active sites, we move on to enzyme inhibition, which is something that affects the rate of enzymatic reaction as well. Enzyme inhibition falls into two main categories: competitive and non-competitive inhibition. The mechanism of competitive inhibition is illustrated here, where the inhibitor occupies the active site of the enzyme, preventing the substrate from binding to the active site to form the enzyme-substrate complex. In the presence of such inhibitors, both substrate and inhibitor hence compete for the same active site. Non-competitive inhibition works differently, where the inhibitor does not compete with the substrate for the active site. Instead, the inhibitor binds to another part of the enzyme called the allosteric site. This form of inhibition is also called allosteric inhibition. Through the secretion of chemicals, the inhibitor modifies the active site of the enzyme, changing its shape such that the substrate no longer can fit into and bind with the active site. This altered active site prevents the formation of enzyme-substrate complex and thus prevents enzymatic reaction. An example of enzyme inhibition is end-product inhibition, which displays an instance of negative feedback. The mechanism of end-product inhibition either follows that of competitive or non-competitive inhibition. As we already know, enzymes react with reactants to produce products. In some cases, the produced products, also referred to as the end product of the metabolic reaction, acts as the inhibitor. End product inhibition follows allosteric inhibition, where the end product attaches itself to the allosteric site. For competitive allosteric inhibition, the end product attaches itself to the allosteric site, such that it blocks the active site and prevents substrates from binding to it. Non-competitive allosteric inhibition is the same as what we previously discussed, where the end product causes a conformational change to the structure of the enzyme, hence altering the active site. The thing with end product inhibition is that it is temporary. Once the product is used up, there will no longer be inhibition, allowing the enzyme to continue forming enzyme-substrate complex again. This brings me to the end of my mind map on proteins and its applications. Do check out my mind map on molecular biology for my introduction to proteins, where I discuss more on protein structure. Anyways, hope you find today's mind map useful and have a good day.